Welcome to Antibody Basics by Biointron, Part 9. In this episode, we'll talk about therapeutics targeting autoimmune diseases. Autoimmune diseases are a global challenge. Autoimmune diseases affect almost 5% of the global population. They represent a diverse group of over 80 conditions targeting various organs and tissues. This dysregulated immune response leads to chronic inflammation and tissue damage, manifesting in a wide range of symptoms across various organs and systems. Examples of these types of diseases include rheumatoid arthritis, lupus, psoriasis, and inflammatory bowel disease. There are various antibody targets in autoimmune diseases. One type are cell surface receptors. These receptors on immune cells can be blocked by antibodies, preventing inflammatory signals. For instance, TNF-alpha receptors are potent inflammatory cytokines implicated in various autoimmune diseases like rheumatoid arthritis, psoriasis, and IBD. Antibody drugs like etanercept and adalimumab bind to TNF receptors, thus preventing TNF-alpha from signaling and thereby reducing inflammation. IL-6 receptors are another key inflammatory cytokine involved in autoimmune processes. Tocilizumab is an example of an antibody drug that targets the IL-6 receptor, blocking both the soluble and membrane-bound IL-6. Thus offering a broader blockade of this inflammatory pathway in diseases like rheumatoid arthritis and systemic lupus erythematosus. Another type of target are cytokines. These are signaling molecules that antibody drugs can neutralize, dampening inflammation. Interleukin-17 plays a crucial role in psoriatic arthritis, promoting inflammation and skin cell proliferation. Secukinumab is an antibody drug that specifically neutralizes IL-17, leading to a significant improvement in symptoms and skin lesions. Another example are B-cell activating factors, which are critical survival factors for B cells, the immune system cells responsible for antibody production. In some autoimmune diseases like systemic lupus erythematosus, excessive B-cell activity contributes to pathology. Belimumab is an antibody drug that targets BAFF, helping to regulate B-cell activity. Co-stimulatory molecules such as CTLA-4 acts as a brake on T-cell activation. In autoimmune diseases, T-cell overactivity can contribute to tissue damage. Ipilimumab is an antibody drug that blocks CTLA-4. This approach is being explored cautiously in autoimmune diseases where a more controlled T-cell activation might be beneficial. In some autoimmune diseases, the target is a specific protein or molecule unique to a particular tissue. For instance, rituximab is an antibody drug that targets CD20, a protein on the surface of mature B cells. This B-cell depletion strategy is used in some autoimmune diseases like rheumatoid arthritis and multiple sclerosis. The complement system is a group of proteins involved in the immune system's inflammatory response. In some autoimmune diseases, dysregulation of the complement system can contribute to tissue damage. Eculizumab is an antibody drug that targets a specific complement protein, C5, which is used to treat a rare autoimmune disease called paroxysmal nocturnal hemoglobinuria, or PNH. As of now, there are 48 antibody drugs for autoimmune diseases approved in at least one regulatory agency worldwide. Humira was approved in 2002 and targets TNF-alpha to treat rheumatoid arthritis. Remicade was approved in 1998, which also targets TNF-alpha, but to also treat Crohn's disease. Benlysta was approved in 2011 to treat systemic lupus erythematosus. The latest drug to be approved last year is Rystiggo, which targets FcRn to treat generalized myasthenia gravis. Let's dive into an example of more specific therapies. B-cell depletion therapies, or BCDTs, reduce autoimmune systems by decreasing the number of B cells and thus the production of autoantibodies, which can mistakenly attack healthy tissues. BCDTs target various molecules on the surface of B cells. For example, CD20, which is the most common target, is a protein present on mature B cells but not on stem cells or plasma cells. CD19 is expressed throughout B cell development, making it a broader target compared to CD20. The B cell activating factor is a molecule that helps B cells survive and mature. One case study example is BCDT in systemic lupus erythematosus. Treating SLE effectively has been difficult due to its heterogeneity, a challenge further highlighted in this bioinformatics era. SLE is characterized by class-switched antibodies to intracellular antigens, especially nuclear antigens. These antibodies can form immune complexes which cause tissue damage in multiple target organs. To improve therapeutic efficacy, Lee et al. Suggests developing combination therapies or to target 2 cytokines with one drug. Biointron focuses on antibody discovery, expression, and optimization services. Our main feature of antibody expression allows your gene sequence to become purified antibodies in only 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. And no antibody means no cost. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca per 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also have several other services, including but not limited to bispecific antibody production, afucosylated antibodies, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression in the mammalian system, and CHOK1 stable cell line generation. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.